Welcome to this video on the glucose-alanine cycle, also known as the Cahill cycle. This is an important metabolic pathway that links amino acid metabolism to energy production, particularly during prolonged fasting, starvation, strenuous exercise, or stressful situations when glycogen stores are depleted and glucose levels are lower. In this video, we will explore the tissues involved, the conditions that activate this cycle, the mechanism, and its physiological significance. Glucose-alanine cycle is an important energy metabolic pathway, also known as Cahill cycle, to honor Dr. George Cahill, who characterized this pathway. This is active when glycogen stores are depleted, glucose levels are lower, fatty acid oxidation and ketone bodies have not started in conditions such as prolonged fasting or starvation. It occurs in three organ systems, including skeletal muscle, circulatory system or blood, and the liver. There are three processes which participate in this cycle. One is glycolysis by providing or by producing pyruvate. Second is transamination to safely dispose of ammonia through formation of alanine. And third is gluconeogenesis by producing glucose. Where does it occur? It occurs in three compartments: the skeletal muscle, blood circulation, and the liver. In the skeletal muscle, glucose is converted into pyruvate, which undergoes transamination to produce alanine with branch chain amino acid. Alanine diffuses through blood circulation, reaches liver, and alanine is converted back again to glucose, which is released into blood circulation, and through blood circulation it reaches different part of body part including skeletal muscle. So it occurs in three compartments: skeletal muscle, blood circulation, and liver. So when is this cycle activated, or when does it occur? This cycle is activated during prolonged fasting, starvation, or strenuous exercise in which glycogen store is depleted and blood glucose levels are lower before significant fatty acid oxidation and ketone body production occur. So let us discuss the mechanism of glucose-alanine cycle. It operates in three compartments: liver, blood, and skeletal muscle. In the skeletal muscle, under these conditions, the skeletal muscle proteins releases branch chain amino acids. These amino acids undergo transamination in presence of alpha ketoglutarate, forming glutamate. So the glutamate carries amino group from all branch chain amino acid through transamination. In the muscle, you also have glycolysis going on, producing pyruvate. Pyruvate and glutamate further undergo transamination, forming alanine. Now in this reaction, glutamate transfer its amino group to pyruvate, which is converted into alanine. So alanine basically carries amino group of branch chain amino acids. Now the remaining carbon skeleton of glutamate is known as alpha ketoglutarate. This can enter into TCA cycle and supply glucose or energy, and also it can join the chain again. Alanine diffuses into the blood being neutral, and it reaches liver where it undergoes further transamination in presence of alpha ketoglutarate. In this transamination, amino group from alanine is transferred to AKG, forming glutamate. So glutamate here in the liver carries amino group of the branch chain amino acid released from skeletal muscle. Now this glutamate releases ammonia, which participate in the urea cycle and remove from the body. So this is the way toxicity is removed. Now you have to take care of energy demand. The carbon skeleton of alanine is pyruvate, which undergoes gluconeogenesis to produce glucose, and this glucose diffuses into blood circulation, reaches in different body parts including the muscle. So this is how this cycle operates in these three compartments. Now you might be thinking why pyruvate does not participate in TCA cycle, why it is undergoing transamination. The reason is because under these conditions the priority is to utilize the skeletal muscle protein, and when muscle proteins are utilized, they release ammonia, which must be neutralized. So to neutralize ammonia, pyruvate undergoes transamination rather than joining the TCA cycle. Regarding the significance, the cycle is extremely important in preventing nitrogen toxicity through formation of alanine by transamination reaction. In addition, it connects protein metabolism to energy metabolism and thereby ensuring proper supply of energy during prolonged starvation or fasting and strenuous exercise. In addition, it maintains blood glucose level, thus supplying glucose to tissues which utilize glucose as the primary energy source. Thank you very much for watching this video. Please like it, share it and subscribe it. Until next time, you take care. Bye bye.